So it's a fantastic pleasure and an honor to sort of wish you happy birthday while it's still your 70th birthday. I've known Pete ever since I came to MIT. He's always been an idol to me as to how he can combine the kindness that you've all heard about and the depth of thinking and the clarity of thinking and just the generosity with which you're sort of sharing your ideas and your time and your insights with all of us. So I've known you as a student, as an undergrad, as a master's, as a PhD student, and now as a colleague and a friend. And just to sort of go through life, watching my family grow and being there so many times with my wife and my kids in this sort of warm embrace that Peter has always given us and our family. So I want to really thank you for this, and thank you for just providing this anchor, this stability in a world of madness, where I know I can always sort of find you at lunch and always get a kind, wise opinion on almost everything. So thank you so much for that. Pete also has been a regular at our salons, and we started organizing these gatherings in the evening at our house with my wife, where we invite luminaries from our community, and oh boy, the list is long, to talk about almost anything and everything. And Pete is a regular there. And what's really funny is that he shows up totally unassuming, and some of our most prestigious and esteemed guests are basically showing up trying to impress Pete because they've known him for decades as this, pioneer in their respective fields. And we are looking up to them, and they show up looking up to him, and Pete is like, well, I vaguely remember interacting with you a little bit a long time ago. So it's very humbling to see how much of an impact you've had on way more people than you can even remember, and to sort of watch that. And something very kind that Pete said about those is that when he first showed up at MIT, he had this image that, Minsky would be having these lunches with everyone and talking about the future of AI. And then he secretly told me, and it's not a secret anymore, it wasn't like that. But now, with these salons, I feel that this is kind of what I had imagined, with all these people coming from so many different disciplines and getting together. So anyway, thank you for all that. Thank you for being such a kind member, above all, and such a wise member of our community. I'm going to treat this as a conference to a bunch of awesome people that I would love to interact with and collaborate with. So what I'm going to do for the remainder of the time is give you a very small glimpse as to what do we actually do when we're not setting up conferences.
By the way, let's all give you a round for setting up an amazing AV.
It's a pleasure.
This is the first time that people applaud before my talk. Let's hope I
just in case we didn't have anything to applaud about.
Exactly. Now at least we're safe. I have two versions, and I haven't quite decided which one to give. So I'm going to give the one with only 70 slides. All right. So what do we do in my lab? So basically, what we try to do is bridge the gap from genetics all the way to therapeutics. So how do we do that? We're basically trying to understand mechanism. And the best way to understand mechanism, since correlation is not causation, is genetics. Because the genetics that we are born with are not influenced by our environment. They're not something that, because I eat more, this genetic variant that I've inherited from my parents, which is a common variant, has now changed. So that is the closest thing that we have in epidemiology to causality, where correlation is much closer to causation. And the promise of genetics is to reveal new disease mechanisms, new target genes, new therapeutics, and to enable precision medicine by pinpointing the molecular basis of all of these traits that we're talking about. And in this particular case, for a body mass index, a measure of obesity, the genetics pointed to a locus that was previously completely unsuspected in this FTO gene, and particularly in the first codon, in the first intron of the FTO gene. The problem, however, is that in 89 common variants in that locus, there was not a single one that was actually perturbing the FTO protein directly. So after more than $1 billion across industry and academia were spent trying to understand the function of the FTO gene, what my group did is pour a little bit of cold water in that party by basically showing the world that it was not the FTO gene at all that was underlying obesity. It was instead two other genes, 1.2 million nucleotides away, which in this resolution is another building over that we're underlying this association. How is that possible? The reason is that FTO is, number one, not an exception. This is true for 93% of disease-associated variants. If you look at genetics and common variants and genome-wide association studies, in 93% of cases, you're going to have non-coding variants without a gene. And the gene could be anywhere. So the reason for that is that these are playing gene regulatory roles. They control the circuitry. They don't control the protein. They affect the circuitry of how this region is regulated. And the target gene does not need to be FTO. But the problem with non-coding variants is that, number one, of course, we don't know the target gene. Number two, we don't even know the causal variant because it could be any of the ones here. We don't know the cell type of action because this variant might be acting in liver, this one might be acting in brain, this one might be acting in muscle. And we don't know the cell type, we don't know the relevant pathway, we don't know the mechanism. So that's what our group tries to do. What we're basically trying to do is figure out what are the genetics telling us about the causal basis of disease and how we translate these genetic findings into mechanistic insights. How? By systematically profiling the non-coding genome, the epigenome, profiling RNA, epigenome, epigenomic modifications at the histone level, at the DNA accessibility level, at the methylation level, that basically tells about the circuitry of each cell. And we're integrating all this data to predict driver genes, regions, and cell types, which we then validate in cell cultures and in mouse models. So I run both a computational lab and an experimental lab. And then, of course, we disseminate the results and we start all over again. So what we're trying to do is basically change these associations with phenotype to mechanistic insights by knowing what are the driver variants, what are the enhancers that they overlap, these distal regulatory control regions, what are the the cell types where these enhancers are acting, what are the upstream regulators, and what are the downstream target genes. In the case of the FTO locus, we like to joke that we wrote a New England Journal paper based on one bit of information in the human genome, namely this T to C variant in this FTO locus. We were able to trace these 89 common variants into a single causal variant and then show that it disrupts an AT-rich binding domain motif bound by this AT-rich interacting domain protein that normally represses these two genes 1.2 million nucleotides away, which we found out they were totally unsuspected to act in metabolism, but they are master regulators of thermogenesis. So these two genes control whether your excess calories in your diet will go to fat or whether they will be burned as heat. And the advantage of figuring out the circuitry is that you can then manipulate the circuitry. We were able to then switch this upstream regulator and these downstream target genes by overexpressing them or down-regulating them and then switch between lean and obese phenotypes. We were even able to use CRISPR-Cas9 genome editing to change that single bit of information in the human genome from C to T and T to C and switch again between lean and obese phenotypes. So the results were just overwhelmingly clear. You could take a fat cell, a primary fat cell, from the adipocytes of the risk individuals and then turn them into healthy adipocytes. These guys are unable to thermogenize, and when you change a single nucleotide, they're completely able to restore thermogenesis. And in mouse, we were able to affect the downstream target genes and show that the mice lose 50% of their body weight. And then when you put them on a high-fat diet, normal mice gain weight, but these mice are unable to gain weight. They just burn calories when they're awake and when they're sleeping. So that's the power of genetics, the fact that once you have the circuitry, you can affect it in a causal kind of way. And knowing causality means you can intervene because, you know, drinking more coffee correlates with living longer. Is it because the people who can afford more coffee and can afford a coffee break can also afford a healthier lifestyle and a, you know, better healthcare system? That's correlation, not causation. Whereas here, we know there's causality. So intervening in causality gives changes that matter. So we don't just do that for one tissue in one region. We do that across hundreds of tissues and hundreds of traits. We basically are able to sort of map all of these complex traits to the tissues where they're likely to act. This can lead to, for example, the brain for intelligence and cognitive performance and math and schizophrenia and neuroticism, etc. But that also has some surprises. For example, Alzheimer's disease maps to immune cells. And we were able to show that indeed, the immune system appears to be at the basis of Alzheimer's disease a few years ago. So we were able to show that immune change in Alzheimer's happened early on, and then much later on do we get neuronal changes. And that actually has completely new therapeutic avenues for Alzheimer's. So that's what we're trying to do. We're basically trying to sort of bridge that gap by not only painting these pictures of as to where variants act, but also profiling the intermediate stages in the disease context. So we can take healthy and diseased individuals, profile their genetics, their epigenomics, their transcriptomics in many different tissues and cell types, and then trace a causal path between them. That allows us to predict the mediating genes and the direction of effect for each of those. So every time we think about a phenotype, we can think about sort of how is it modulated at the molecular level. And more, we're doing this at the single cell level. We basically now have new grants to profile at the single cell level Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, AD and psychosis, schizophrenia, bipolar, Down syndrome, autism, depression, suicide, PTSD, and aging. And for each of those cases, we're looking at dozens of individuals, dozens of traits, all of the major cell types, sometimes multiple regions in the brain, and many times both RNA and DNA accessibility. What are we finding? We're finding that we can paint these cell types even across health and disease individuals. The primary axis of variation appears to be the cell type. And within that primary axis of variation, the secondary is whether individuals that those cells came from were healthy or sick. In this particular case, I'm showing you 84,000 cells across 48 individuals. Half have AD, the other half have have no AD, the other half have either early or late stage AD, and every single dot is one cell from these individuals. And you can see that the primary clustering is indeed cell type, but within it, these cells come from sick individuals and these cells come from healthy individuals. So we were able to subdivide these cells into the major cell types and then subdivide each major cell type into clusters. And what we found was striking. Every single major cell type showed two major axes, one for high amyloid, one for low amyloid, one for late Braak stages, one for early Braak stages, one for high cognitive decline, one for early cognitive decline, one for high AD, one for low AD. So even though the genetic variants primarily localized in microglia, the expression changes are found in every cell type. And that's quite important. The second thing that was quite striking is that women have much more sick cells than men. So the cellular signatures of expression in Alzheimer's are much more common for women than for men, which correlates with the higher prevalence of Alzheimer's in women. But in our cohort, we had actually tried to match both the cognitive and the pathological signatures of AD between men and women. So that in fact suggested to us that women are in fact more resilient to AD, even though they have the same amount of gene expression change. It also pinpointed pathways that are dramatically different between men and women. So men are able to upregulate myelination pathways after AD strikes, but women are not. And in fact, when we look at the pathology, when we actually look at the radiology results for these individuals, we find a lot more white matter loss for women. So that suggests that somehow men are able to protect their neurons by myelinating them, but women are not. Again, completely different therapeutic avenues are coming out of this data. And of course, we're doing things in Huntington's. For example, we're finding that the single cell profiles in Huntington's are dramatically different, specifically for this indirect pathway spiny projection neurons that appear to be merging their signatures in HD versus controls, and that has a lot of implications for how to treat those. In schizophrenia and bipolar, we're able to find what are the major cell types that are changing in each of the cell types, and they differ by cell type. We're able to now partition these single cell effects at the genetic level to basically ask what are the EQTL, the expression quantitative trait locus effects, that are associated with Alzheimer's, but now which cell type do they act in to change expression? And again, you see here the microglia appear to be sort of the primary place where these genetic variants are acting. And of course, after that, changes are happening in all of the cell types. We're also able to look at blood biomarkers of disease. In this particular case, we're finding that about 100 genes show differential epigenomic signatures in AD versus non-AD individuals, which has, again, a lot of implications for prognosis. Some of those genes are in fact falling in macrophage pathways, which again mirror the effect in the microglia inside the brain. Some of that could be due to common signatures between the CD14 plus monocytes in both blood and brain. Some of in Alzheimer's, and therefore that the macrophages are indirectly having an effect. And the stuff that's perhaps of the greatest interest to this audience is that we were able to subdivide this major classification of AD as a monolithic disease into subphenotypes of A beta analytic plaques, tau and neurofibrillary tangles, as well as neuroinflammation. And when you do that, you can actually find that these partitions the genome according to the regions whose methylation is associated with specific combination of these phenotypes. So that's extremely important when you're dealing with a clinical record. It basically says that if you have combinations of subphenotypes, you can perhaps partition the patients to gain insight into the distinct biological pathways associated with different combinations of phenotypes. So that's what led us to start studying MIMIC and EHRs in collaboration with Mayo and partners to basically start looking at the comorbidity patterns across a large number of phenotypes to start building these hierarchical models for how we can understand patient classification by combining lab tests, CD9 codes, prescriptions, DRG codes, and even the doctor note free text to basically build these hierarchical models that now allows to classify patients based on these multimodal phenotypes shown here according to those colors. So every one of these classes is defined by a combination of variables that is in fact implicating multiple data modalities. And that allows us to now start completing the clinical record. We can basically go back into MIMIC and say, hey, these data are a little sparse because, of course, they don't bother billing you for every single related disorder. Can we start filling in these data and sort of completing it, in some cases correcting it, in places where the algorithms are clearly pinpointing that that patient belongs in a different category and these variables seem to be off. So applying this methodology in the context of PTSD in collaboration with Nikos Daskalakis over at McLean Hospital, to basically subdivide patients according to their combinations of phenotypes across many different phenotypic variables, and then combining genetics from those patients to actually predict expression levels in each of a dozen different tissues to basically see what correlates with the genetics of each group and therefore the predicted gene expression impact of those genetic differences in order to actually assess what are the tissues that likely underlie each of these variables. We can now extend that to polygenic risk scores. Even in a cohort where we do not have the phenotypic information, we can actually construct the phenotypic information from polygenic risk scores, from the genetics of these individuals, predict gene expression patterns at the tissue level, deconvolve that at the single cell level, and start doing correlations between what are the cell types that appear to be underlying which disorders, just by having genetics in a cohort, which is actually a little mind-boggling. We can then use all this information integration to start prioritizing driver genes and the cell types where they appear to be active, using this mediation framework that basically tells us what are the intermediate variables on the way to disease, and what are the cell types, the tissues, the genes, the regions. And then when we do that, we can actually prioritize a large number of genes in schizophrenia, in bipolar, and each of these disorders. And we now have an ample program for dissecting these experimentally. So Maria Kus is a postdoc in my lab who's now, actually generating these images of synapse, connectivity across time and across conditions at different days and ages. In collaboration with Litway Tsai's lab, we now have an experimental component at the Piper Institute where we're basically creating these programmable CRISPR-Cas9 constructs to modulate either the epigenome of these cells by using dCas9 with an activating or a repressive domain, or Cas9 with genome editing, or simply Cas9 to cut the genome and deactivate genes or promoter regions. And we're carrying these out in IPS labs, which we then differentiate into neurons, microglia, astrocytes, oligodendrocytes, to basically understand in what cell type do these changes actually manifest in a causative, interventional kind of way. In collaboration with Kevin Egan, we're trying to multiplex these experiments to basically carry out 384 of these experiments at a time. And we've also developed assays that allow us to profile 7 million regions in a single experiment to basically profile where are these genetic signals and genomic signals coming from, what are the driver nucleotides, and in this particular case, we can pinpoint individual motifs, individual conserved elements, and sort of drive that. And then in collaboration with Andreas Penning, a former postdoc in my lab, now he's his own lab at CMU, as part of the Cure Alzheimer's Consortium, we're basically now modulating hundreds of these SNPs at a time, inserting them into these constructs within the brains of mice, basically look at the in vivo impact of these single nucleotide perturbations at the enhancer level and ultimately at the neuronal connectivity level and so on and so forth. So this is a huge collaboration with many, many people. Several of them have now grown off to their own labs. So, constantly looking for collaborations, constantly looking for new students and postdocs, and that's sort of a little bit of where we're coming from. So basically, the key takeaway that I wanted to leave you with is that we're basically trying to bridge that gap to basically make this genetics to disease gap much, much smaller by understanding what are the causal pathways that are implicating the disease, and the fact that we can manipulate those and actually lead to the therapeutic. That's all. And I'll be happy to take a question.
Quick preview.
So,
yes,
one of the collaborators there was from McLean Hospital.
Yes.
How do they react to that in terms of they say, wow, we should start using this? What's their thinking?
So there's actually a lot of collaborators that we have in McLean. So 2 of them are actually clinical fellows who are in my lab. I'm the mentor for both their K awards. So they spend a day a week in my lab, and they call in for video meetings all the time. And they actually see patients. They collect samples. They do mouse experiments. So these are not your typical psychiatrists of, oh yeah, I'm just seeing patients. These are actually, working on the molecular basis. So maybe I'm not dealing with the traditional doctors, but they are fully on board with the whole thing. Basically, they're basically saying, let's understand causality, let's understand intervention, and now we're doing some programs where we're actually going to be profiling brains of individuals on different drugs to basically understand what are the molecular consequences of using these drugs between, cases and controls. Of course, there's a lot of confounding because people with similar phenotypes are given similar prescriptions, and it's a little unethical to sort of do a clinical trial, or at least a randomized trial in that case, when you think that the best standard of care is X and Y and Z for a particular trait. But we are doing those studies right now. So I'm a little biased, but at least the reception from the true doctors has been overwhelmingly scientific and genomic.
Those are absolutely true doctors. You answered my question, but really my question was, were they just interested in the science, or were they also looking for some translational impact?
I think the translational impact goal is always there, but we're all very realistic about how fast it can happen. So I'm hoping that, we will gain these insights, but I think the clinical trials for this will take many, many years. I feel that, however, at a very small scale, we have the potential to impact, because the doctors that I'm dealing with have actually the freedom to sort of prescribe this drug or that drug. So, I don't know what the IRB and ethical approval will be for sort of having science that we do that might be in some cases unpublished influence those decisions. But if both of them fall within the standard of care, I don't see the harm in that. But again, I'm not into this world of ethics as deeply to fully understand the consequences.
So Manoli, I was really struck about a dozen years ago by a talk that Joe Von Ventry, who was the Harvard head of the HST program at the time, gave. So it was to a group of biomedical researchers, and he started off being very complimentary. He said, "You guys are so clever. You look at all this data. And you come up with these really interesting
trouble ahead."
Ideas."
Yeah.
And then his punchline was, "Stop doing that." And his argument was that every time he read a paper along these lines, he had to go out and hire a postdoc and do a bunch of experiments to see whether that insightful observation actually was real. So how do you avoid that problem? Because you guys, before you go out and start modifying genes in mice, you develop ideas by doing data analysis. But what is it about your approach that wouldn't suffer the same problem that Joe was talking about?
So causality, I think, is where it all lies. In correlation analysis, you can get fooled so easily. By using the genome, you have a unidirectional arrow coming out of the genetics. So as soon as you can play these statistical tricks to get that unidirectional arrow to now propagate to various other intermediate phenotypes, you can sort of have much more confidence in your result.
But the causality only becomes obvious when you do the experiments.
I agree.
Just the fact that
I agree, but we're guiding.
Involved,
yeah.
One of your examples showed that it wasn't that gene; it was some other gene that.
Yeah.
Was
so the very first example that I gave, it's kind of funny so we're having this industry academia joint gathering once a year, and I was one of the speakers in one of the years. And the person who introduced me as the person who wrote the most depressing paper of 2015. And the reason for that was this FTO paper that I mentioned in the New England Journal. And the reason for that is that every single time they find 5 genes under the lamppost, basically namely where the genetic locus is, they're like, oh, this one's complicated, it has 5 genes. Which of the 5 could it be? And what we showed is that in this one case where the whole field was sure that we had the gene, it wasn't under the lamppost at all. It was way, way far away. And basically the message that I'm trying to give is don't look at simply where the genetic variants are located. Look at what they're actually doing. Because everybody goes with simply, oh, it's right next to that gene, so that must be the gene. And what we're looking at is what is the full circuitry? And that is what gives you sort of the right target gene. So in my view, just looking at where the genetic variants are sitting doesn't tell you anything. It's kind of like saying, you know, oh, there's a, I don't know, spill in the parking lot, but you don't know which car was going through when the spill happened. And therefore sort of being able to trace it to where that thing is actually connected is what was missing. I hope that answers your question. All right, great, thank you.
Thank you.